We're talking about the transport of gases in this lecture, and the two gases we're concerned about are oxygen and CO2. And there's different ways that these transport or find their way into the blood. Okay, we'll start off with oxygen and then we'll jump into the CO2 story afterward. Now, oxygen, what happens once it passes through the alveolus across the respiratory membrane into the capillaries, the pulmonary capillaries, about 1.5% of it's dissolved in the plasma. So, in essence, there isn't a lot of solubility there, right? There's this very small amount of oxygen that is willing to just go right into your plasma and stay there. Fortunately, though, you have this molecule, which is known as hemoglobin, that actively binds oxygen, and this is what greatly increases the solubility of oxygen. This is also the reason why you don't have a whole bunch of nitrogen inside your blood, because we don't have a big protein in our blood that preferentially grabs onto nitrogen. But we do have one that preferentially grabs onto oxygen, so it greatly increases its solubility. Here's what hemoglobin looks like. It's a quaternary protein, which means it has 4 individual proteins that are all associated with one another. And each one of those portions brings with it a special part of the molecule, which is called a heme, as we saw in the blood chapter. And on each heme, see if you can recall, there's a special type of heavy metal that is known as iron, and these iron atoms are the ones that are highly attracted to oxygen, and that's why oxygen wants to go there. Now, the strength or the affinity with which oxygen wants to stay on the chair will vary depending on the partial pressure of the gas. So there is what you could create an oxygen hemoglobin dissociation curve, which shows this principle. This is what a standard oxygen hemoglobin dissociation curve will look like. And basically what you're seeing on the left is how much, what is the oxygen saturation of hemoglobin. So this, think about it being this way. At 100% saturation, it means on average, all of the 4 chairs, so all of the 4 heme groups on a molecule of hemoglobin, on average across all of the hemoglobin molecules, all the chairs are occupied and there's no more room at the end. All hemoglobin is saturated with oxygen. That would be 100% hemoglobin saturation. On the flip side, let's say you assess all of the hemoglobin molecules in the entire blood supply, and on average only 3 of the 4 chairs are ever occupied by oxygen. Well, that would be a saturation of about 75%, meaning you could still fit another 25% of the capacity of oxygen onto the molecules. And then similarly, if it was a hemoglobin saturation of 50%, that would mean that roughly speaking, on average, only half of the chairs are full of oxygen. So think about hemoglobin being like a shuttle with 4 chairs on it, and oxygen can jump on or jump off. And sometimes oxygen wants to jump off more easily than at other times, and this would be called the affinity for oxygen to dissociate. If there's a strong affinity for oxygen possessed by hemoglobin, it means that the oxygen is reluctant. It doesn't want to jump off the chair. If there's a low affinity for oxygen, that means that oxygen doesn't really want to stay on the ride. It wants to jump off. Okay, and that affinity will change, and that's why we have a different shape of this curve. So let me explain a little bit about what's going on here. How much saturation we see in hemoglobin depends principally on the partial pressure of oxygen. So in the lungs, we said there's about a partial pressure of oxygen of 100. Just to go back here, we're getting that number right from here. Okay, we said there's about 100 millimeters of mercury partial pressure of oxygen in the lungs. So that's what you're seeing on this graph here. And when you do have those conditions where you have 100 millimeters of mercury of partial pressure of oxygen, the hemoglobin of blood will be nearly 100% saturated, meaning anywhere you look, if you pull the hemoglobin molecule out, it's going to have 4 of the chairs occupied with 4 molecules of oxygen. It's totally saturated. There's no more empty chairs for oxygen. When blood travels to a place such as the tissues, the peripheral tissues of the body that have 40 millimeters of mercury partial pressure, again, the number I get, where I got that number is right here. We said that at the tissues it's about 40. Okay, then if I was to look at all of the hemoglobin molecules, on average, this would be 70. So it's about 75% saturation. So in other words, this is something interesting to appreciate here. This is the normal flux that happens between external and internal respiration. The blood goes to the lungs, it gets full of oxygen, and even after the blood is leaving the tissues, at the site of the tissues, this is where the blood is going to be most oxygen depleted. But even then, still look at this, 75% still of oxygen remains. So there's sort of a misconception here that I think students get caught up on when they say that arterial blood is oxygenated and venous blood is deoxygenated. I say rubbish because even deoxygenated blood still has 75% of all of the oxygen in it. I mean, it's still loaded with oxygen. So this is a good thing, meaning you have way above and beyond the amount of oxygen that you need, which also means you have a very large reservoir of oxygen remaining above and beyond what you typically use in a normal physiological day-to-day situation. All right. So you've got per when your blood is most saturated, it has about 20 milliliters of oxygen per 100 mils of blood. And then you only lose about 5 mils of that oxygen, but you still have 15 mils of oxygen dissolved in your 100 milliliters of blood. So there's a lot there, okay? So that's one important feature to note. Now, you're going to see, though, the affinity decreases as you get further down in the partial pressures. The steeper the gradient, the more the affinity decreases. So here, there's actually still a pretty high affinity for oxygen, right? Because this is a pretty flat part of the curve, which basically means, like, I can get, for example, to a place where there's maybe only 70 millimeters of mercury, a partial pressure oxygen. So that's actually, a pretty big dive down from 100 to go from 100 to 75, and I lose almost 0 oxygen because oxygen doesn't want to get off. It likes being on the chair of hemoglobin. And even at a place in the body where I only have 70 millimeters of mercury of partial pressure oxygen, the oxygen's not going to really go there. A very little amount of it will go there. Now, as we get closer to 40, now oxygen's saying, okay, I'm happy to get off the hemoglobin chair. I can see there's a greater need for oxygen here. And so now you start to see the shape of this curve become more steep. With every decreasing amount of partial pressure of oxygen in the tissues, there's a greater degree of oxygen that wants to jump off of the hemoglobin. And then from here, now you see a steep decrease in the affinity. At this point, once you get beyond 40 and less, now oxygen is very eager to jump off. So there's a change that happens to the hemoglobin molecule. The more oxygen gets off, the more rapidly the oxygen wants to get off. It's like peer pressure, right? You see, you're sitting on the oxygen train. Some of your buddies get off the train. You're like, oh, maybe I should get off the train. And so now everybody's jumping off the train. That's sort of what's going on here with hemoglobin. When the train's full, nobody really wants to get off. But as people start jumping off, it becomes less likely that people will stay on. And so they jump off more vigorously, which is good. Because if you get to places like this in your body, any place in your body where you only have 30 millimeters of mercury, this would be like the condition in really active skeletal muscle, or like sometimes in a skeletal muscle that's really active and using oxygen intensely, you might get even as low to a partial pressure of 20. Well, you need a much more rapid diffusion of oxygen to those regions. And so if the blood does pass by a capillary bed in a tissue such as that, oxygen at that point will be very eager to jump off. And you've got a lot of it still remaining. Remember, like here you still have 75% of it remaining. and quickly you could get down to 25%, and in those higher active tissues. So the affinity that oxygen has for hemoglobin changes depending on the needs of the body. When there's plenty of oxygen around, oxygen is less eager to jump off of hemoglobin. When there's not much oxygen around at the tissues, it's much more eager to jump off of hemoglobin and get into the tissue. But generally, in a day-to-day situation here, you're looking at only getting rid of 25% of the hemoglobin, and generally 75% of it will still remain in the blood, even in the deoxygenated, quote-unquote, state. So for that reason, I usually like to call venous blood oxygen poor versus deoxygenated, because it does still have quite a bit of oxygen in it. And how saturated hemoglobin is will have a bearing on its color. It actually undergoes conformational changes. It changes its shape depending on how saturated it is. And when it's really saturated, it's bright red. And when it's at about 75% capacity, it turns more of a maroon color, which we see as being more of a bluish, purplish color. So when you look at your veins, for example, on your hands and your feet that are more superficial, they will look a little bit more maroon or blue, indicating that the blood that's in those is more of this situation, having a hemoglobin saturation of about 75%. Now, there are some things that can change that curve. One of them is the partial pressures. Okay, we saw that hemoglobin saturation is going to be affected predominantly by the partial pressures of the tissue that are surrounding the blood. But that's not the only thing. There's actually a molecule called bisphosphoglycerate, or BPG, and this is a byproduct of glycolysis. And red blood cells do not have mitochondria in them, so their only way of making energy is glycolysis, which is good for them because they don't really do anything except just sit around and carry oxygen. Okay, they don't have to they're not making a lot of stuff, they're not regulating a lot of stuff. They're really, at the end of the day, just big old sacks of hemoglobin. And so they don't require heavy-duty energy-making machinery. Glycolysis is fine for them, and they make BPG in that process. Now, what happens to it is BPG will promote the dissociation of oxygen. So if BPG is around, it encourages oxygen to get off the chair and go to the tissues where it's needed. So it lessens the affinity of hemoglobin to oxygen. Now, here's the problem. When you give blood and you put your blood in a blood bank, the cells have a sort of a limited shelf life because there's only so much glycolysis that can be made, and once the glycolysis has finished up, then BPG starts to go away, and as BPG starts to deplete, it can be the case that when there's not enough BPG around, the oxygen is actually going to want to stay on the hemoglobin. So, for example, if you gave somebody a bag of blood that was too old and had not enough BPG in it, it wouldn't do them any good, honestly. You would be giving them hemoglobin that never wants to loosen its grip off of oxygen molecules, and that wouldn't be great because the oxygen would certainly go onto the hemoglobin, very good, but it wouldn't ever want to jump off. It takes a little bit of the BPG to encourage the oxygen molecules to jump off. And if the oxygen molecules don't ever jump off of the shuttle, they're not going to go to the tissues, into the cells that need them. Okay, so that's an important consideration. That is why blood that is donated has a shelf life, so to speak, to it. Now, pH will also affect the affinity of hemoglobin to oxygen. And I might ask you a question here. Do you think that lowering the pH or raising the pH will increase or decrease the affinity of hemoglobin to oxygen? I bet you could figure that out just based upon logic. And I will say it's a pretty logical answer. Would you need oxygen to get off of hemoglobin more or less rapidly under conditions of acidosis, let's say? I'll show you a curve here that gives the answer. But generally, if you have a more acidic pH, it encourages oxygen to jump off. It means it lessens the affinity of hemoglobin to oxygen. And similarly, temperature. If you have higher temperatures, it encourages oxygen to let go of hemoglobin and then enter its tissues. And both of these are a good thing. Here's a couple of charts that show what I mean. So, for example, let's say you're assessing the partial pressure of oxygen at the level of the tissues where the blood is going, okay? The curve shifts depending on the pH. See this? So at a pH of 7.2, this is an acidic pH. At a partial pressure of 40, you would have only about 60% hemoglobin saturation, meaning only about half of the chairs have oxygen. Oxygen is jumped off readily, okay? Whereas at a pH of 7.6, just a little higher, now you have 80% of the chairs occupied. So there's more oxygen that is bound to hemoglobin when the pH is greater, and there's less oxygen that's bound to the hemoglobin when the pH is less. And this is good because acidic pHs generally mean that there's too much activity on behalf of the cells. When cells are active, they create CO2. The CO2 lowers the pH, and so a pH that's less would mean that probably cells are hyperactive, and in those conditions, they're going to need more oxygen. So it would make sense that in a more acidic environment, you would be able to take the oxygen off of the hemoglobin more rapidly so that it could go into the tissues and be used to make energy. Similarly with temperature. Okay, big difference here. Look at this. We come up at the level of the tissues. If the body was 43 degrees Celsius, you would have only close to less than half, or close to half of the hemoglobin saturated. Whereas if you lowered that temperature, like in a refrigerator, let's say, hemoglobin's not going to let go of oxygen at all. So the more you warm up the blood, the more willing hemoglobin is willing. The more willing hemoglobin is, or the more hemoglobin is willing to release its oxygen. All right, so hemoglobin plays a big, important role in carrying oxygen. Now, what happens to carbon dioxide? It's a little bit of a different story. Carbon dioxide is more readily soluble in the blood, and so 10% of it actually would dissolve into the plasma. This is different than 1.5%, which is the case for oxygen. Okay, so much more is going to just, from the tissues, just dissolve straight into the plasma, the fluid parts of blood. As that blood passes through the capillaries of the tissues of the body, you have CO2 entering. Now, 70% of the CO2 then is going to dissolve into the plasma, but then it's probably going to be converted into bicarbonate ion and it will be stored within the red blood cells. And that is a really important chemical reaction that looks something like this. If you take CO2 and you add it to the blood, what happens is that the CO2 in the water inside the plasma of the blood reacts to create a substance known as carbonic acid. The enzyme that facilitates this reaction is called carbonic anhydrase, and it's found inside your blood. Okay, so if you have CO2 accumulating, it drives the reaction this way. Note that these arrows are both directions, so this can go either way depending on the reaction according to Le Chatelier's. Is that the right name? Le Chatelier? Yeah. It's been a long time since I've taken chemistry, but according to that principle, wherever you stack either the reactants or the products, it's going to drive the reaction the corresponding way. So if we're adding CO2 to this side of the equation, for example, it's going to drive the reaction this way. If we were stacking this side of the equation, it would drive the equation the other way. Well, carbonic acid will then, if it continues through the reaction, it's going to dissociate into hydrogen ion and bicarbonate ion. That's what this ion is here, HCO3-. And that's going to stay inside of the red blood cells. So much of the CO2 the way that it's transported, 70% of it, it's transported in a funky way as this molecule bicarbonate ion, which came from when we combined CO2 with water. Ultimately, it created the bicarbonate ion. And so if you were assessing blood, let's say, and you saw that there was a high hydrogen ion concentration, or you even saw there's a high bicarbonate ion concentration, but generally this is what the chemoreceptors would measure. If you were measuring blood and saw a high hydrogen ion concentration, that would be a signal that there's a lot of CO2 in that blood, and that would tell you that you need to start breathing more, because every time you breathe you get rid of CO2. And so if there's too much in the blood, it means that it's not getting out as it should. It means it's either being produced in too high of a quantity, or you're not getting rid of it as quick as you should, or perhaps both. You're becoming acidotic because there's hydrogen ions accumulating in there. And then the rest of it, about 20%, is bound to hemoglobin, although we have to be careful here. Oxygen is bound, see if you can remember, where is the oxygen bound on the hemoglobin? It's bound to the heme groups, specifically the iron. And the CO2 then is actually bound to the amino acid bits. So if I go back to this picture of hemoglobin, oxygen rides on the iron, so these very small little portions here, and the CO2, it hangs around the globular portions, the amino acid portions of the hemoglobin molecule. And so there's less to do with chemistry here about hemoglobin's affinity for CO2. We don't really generally concern ourselves with pH or temperature in terms of how CO2 is transported, because a lion's share of it is transported just into the blood. And so then imagine if the blood is passing through across the alveolus, then what happens is that CO2 starts leaving because it diffuses out and into the alveolus. And as the CO2 leaves, it actually drives the reaction back this way. So when CO2 is being onboarded onto the cells at the level of the cells and the tissues of the body, the CO2 is getting pumped in, that's driving the reaction this way. But once you get to the lungs and CO2 wants to jump out of the blood, then it drives the reaction this way. And then what you have is bicarbonate ion and hydrogen ion coming together, forming carbonic acid, and then releasing the CO2 as a gas form that can then bubble out of solution, accumulate in your lungs, and you breathe it out. All right, that's the place that we will stop here on the respiratory system. And I hope this gives you greater appreciation for all those breaths that you take every single day, what's happening to those gases, how it's happening, and why it's important.